Experiments in chick suggest that the primitive streak is induced by cell-cell interactions at the caudal end of the embryonic disc. Although the exact tissue interactions are disputed, it's clear that extraembryonic tissues induce the adjacent epiblast to form the primitive streak, and that this process of induction continues as the extraembryonic endoderm migrates from the caudal to the cranial. Mis-expression studies, including gain of function and the loss of function experiments, suggest that TGF-beta and Wnt family members induce the primitive streak. In chick, VG1 is in conjunction with Wnt8A induces the epiblast to express another TGF-beta family member, nodal. VG1 is one TGF-beta family member, nodal in turn, along with FGF8, causes epiblast cells to de-epithelialize and form the primitive streak. Finally, inhibition of the endogenous BMP signaling also seems to be required for primitive streak formation. In mouse, Wnt3 and its downstream target brachyury are expressed in both future cranial and caudal pre-streak epiblast. During subsequent development, Wnt3 is downregulated cranially by signals from the specialized region of extraembryonic endoderm called the anterior visceral endoderm, and is upregulated caudally. Finally, expression of the Wnt3, brachyury, and the nodal becomes consolidated within the primitive streak. Loss of function mutations of these genes expressed by the anterior visceral endoderm, for example, CER2 and LEF2Y, both inhibitors of TGF-beta and Wnt signaling, result in the formation of extra primitive streaks. Moreover, embryos with the loss of function mutations of nodal fail to form primitive streak. Further studies in mouse revealed that the formation of the primitive streak involves the signaling of the TGF-beta family members from extraembryonic tissues. Studies in chick have revealed the cellular basis of primitive streak formation. 4 major processes are involved, including oriented cell division, oriented cell migration, and convergent extension and progressive delamination from the epiblast. As covered above, during the formation of the primitive streak, cells are induced from the epiblast by the caudal extraembryonic region. As induction occurs, these cells delaminate from the epiblast and migrate cranially and medially. Analysis of labeled clones of cells show that cells are displaced mainly cranially as they undergo division, suggesting that their division plane is preferentially oriented in the medial lateral plane, so that daughters are displaced cranially. As extraembryonic endoderm migrates cranially, progressively more cranial epiblast cells along the midline are induced to delaminate, extending the cranial end of the primitive streak more cranially. Finally, cells within the forming streak merge medially, and subsequently the streak extends cranial-caudally to accommodate the merging cells. Thus, convergent extension contributes to the later aspects of the primitive streak formation and elongation. As we know, human embryonic stem cells have the ability to differentiate into all human cells. However, controlling the differentiation has always been a challenge. By activation of TGF-beta pathway, some scientists investigated the direct differentiation of human embryonic stem cells to primitive streak-like cells and further into pure mesoderm and mesenchymal-like cells. Taken together, these findings have demonstrated the protocol for directing differentiation of human embryonic stem cells through the gastrulation step of primitive streak formation into a definitive mesendodermal stage. From the normal embryonic development, we can get a lot of inspiration for scientific research.